Hello and welcome back. Today we begin our unit on biological information and this idea of a central dogma where information is passed from DNA to RNA to proteins, and that's where the genes that are stored as DNA actually get expressed. So the proteins are what make you and me, you and me. So the color of our hair, the color of our eyes, our metabolism, all the enzymes that carry out reactions in our body are expressed as proteins, and those start as genes in the DNA. So we're going to look at the transfer of information from our DNA, from our genes, to what actually gets expressed as proteins. So this idea of the central dogma, what it does is it outlines, give me a second here we go, it outlines the flow of information in biological systems. So we're going to break it down into four parts. The first one that we're going to look at right now is a quick background of DNA and the double helix. What is it about the structure that allows it to carry information? Then we're going to look at the second one will be DNA replication, and then we'll look at transcription, and then finally translation, so the processes that are involved in this. So let's begin with our look at DNA. Oops, real quick again, the central dogma. So here again, just a flow chart that shows DNA. And what we'll look at with DNA is the structure, but then how can it replicate itself? Every time a cell divides, every time you get a scratch, every time you slough off a little bit of skin cell, bite the inside of your mouth, something like that, cells have to be replaced. And in order to replace them, we've got to copy the DNA. So how does DNA copy itself? So we'll look at that process first. And then we go and we look at transcription. So transcription is copying. So hopefully you are transcribing these notes right now. You're just copying it from one form to the next, and it stays in the same language. And in this case, that language is nucleic acids. So it goes from DNA to RNA. So we know there are some differences between those, but it's still nucleic acid. And finally, we look at translation. So here's where we copy it to a new language. So here's where we go from nucleic acid to protein. So if any of you are in Latin or French or Spanish, you may have to do translations from one language to another. And that's really what's happening in translation. We're taking this information and we're copying it from a nucleic acid into a protein. We're changing languages. So now let's start and look at how the DNA is actually put together. So we've seen this structure before when we talked about biomolecules. So this is a nucleotide. It's the monomer of nucleic acids. And it's got three main parts. Up here on the upper right in the green, it's got a nitrogen base, it's got a 5-carbon sugar, and it's got a phosphate. Now what I want you to focus on right now is look at the sugar. And it's a 5-carbon. And they're not shown, but there's a carbon here, 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 and here. Now those carbons are numbered. 1, 2, 3, 4, 5, and often they're called prime. Now the two that I want you to focus on are the 3' and the 5'. Those are going to be the most important. And I'm going to draw in off the 3'. There is a hydroxyl group that's not shown. That's going to be very important because what do we know about OH groups? Well, we know that if there is another H available, they can form a condensation reaction, and we can form a linkage. In this case, it's a phosphodiester bond. So this 3' carbon with the OH is going to allow us to link these nucleotides together into a polymer of nucleic acid. So you need to recognize the nucleotide structure and know that there are 5 carbons. And when we talk about 3' and 5', what we're going to be dealing with is it the 3rd carbon right there at the bottom, or is it the 5th carbon here at the top that has the phosphate attached to it? Now, because of the 3' and 5' structure, there is that free OH group on the 3' end. The nucleotides are always going to be added in a 5' to 3' direction. And that's going to be very important. So what that means is right down here on the 3', there is an OH, so we can always add to that. So the chain, the polymer of nucleotides, is always going to be extended in the 5' to 3' direction. And if you look in the lower right-hand corner, we've got our individual nucleotide, and again, there's the 3' and the 5' and why those are so important. So what that means is when we have this growing strand of nucleic acid, RNA, or DNA, we're always going to extend it only in one direction. This phosphate group at the 5' doesn't have the OH for a condensation reaction to take place. So when we look at it, and here's another cartoon version of it, if we look in the upper left-hand corner here, we can see, yep, we are always going to add a new nucleotide to the 3' end. That phosphodiester bond forms. Fancy word for a condensation reaction with nucleotides. And we get, in this case, a dinucleotide. And we can add another to this end right here, and another, and another. And what happens is we get this chain that grows in the 5' to 3'. And in the diagram shown here, we've got 1, 2, 3, 4 nucleotides that have been linked together. So there's our growing nucleotide. So remember, the chain is always going to grow 5' to 3'. Now, what does the structure of DNA actually look like? Well, DNA, as you may remember, is a double helix. And what that means is there are 2 strands of it, and they're wound around each other, kind of like a spiral staircase. So we've got this sugar phosphate background, kind of the ribbon structure shown here. And there's 4 different bases. There's adenine and thymine, A and T, and then there's cytosine and guanine, A and G. Now, when we talk about RNA, you've got to remember that we replace the thymine with uracil, but for our discussion right now, we're going to look at these 4 bases and concentrate just on DNA. So there's the double helix model. Now, let's take the spiral out of it. If we look at the diagram on the left here, here is a strand of DNA. It's a double helix, so it's got 2 strands. And there is a sugar phosphate backbone on each side, so that's kind of like the outsides of the ladder. So we've got here and here. There's the sugar phosphate backbone. We have nitrogen bases in the middle. They form the rungs of the ladder. So if we will go across here and here, right down there, and they are bonded together with hydrogen bonds in the middle to form kind of the rungs of the ladder. And what this structure gives us is allows that there's always complementary base pairings. So what you've got to remember is, just like the diagram, adenine will always and only bond with thymine. And cytosine will always and only bind with guanine. So that's what complementary base pairing means. And you can see in the diagram here that it doesn't matter which side they're on, but if you've got an A, it'll always be paired with a T, and if you've got a G, it will always be paired with a C. The other thing that means is that when we have a double strand, those 2 strands are what we call anti-parallel. So one of the strands, the one on the left, as you can see, runs from the top 5' down to the 3'. The opposite side has to be anti-parallel. It runs from the bottom 5' up to 3', so they're anti-parallel. So those are 2 things about DNA that you've got to remember. There's complementary base pairing, and that they do run anti-parallel to each other. Now, why are they complementary? If we look at this, we can see that adenine will always only pair with thymine. And the reason for that is it forms 2 hydrogen bonds, and guanine only forms with cytosine, and it has 3 hydrogen bonds. They go together. The other reason is if we look at the nitrogen bases, we can see that adenine and guanine are both double ring structures here, adenine and guanine, and they're called purines, okay? And we're going to get into that in more detail in class, but for right now, just know that adenine and guanine are double rings, thymine and cytosine are single rings, and they are pyrimidines. And what that means is when they go together, we want to have an equidistant between the rungs of the ladder. So we always want to have a double ring with a single ring, so we get 3 rings across. So that's why adenine and guanine won't go together, because that would give us 4 rings, and that would be too wide. Thymine and cytosine would be only 2 rings, and that would be too narrow. So we always pair adenine with thymine, guanine with cytosine. That added to the fact that thymine and adenine have a double bond and cytosine and guanine have a triple bond means that T always goes with A, C always goes with G. And here's what happens when you put all of that together. Here is our anti-parallel double strand of DNA. So you can see the left hand goes from 5' in the upper left to 3'. The right hand goes opposite. It starts in the lower right and goes up. So we've got our, sugar phosphate backbones. We have got our nitrogen bases in the middle forming the rungs of the ladder. And we always have thymine with adenine, as the first group shows. We always have guanine with cytosine. Doesn't matter which side is which. Here's cytosine on the left and guanine on the right. And so there is the basics of the DNA structure and how the nucleotides fit together to give us this double helix structure. And the next time we get together, we'll talk about how we can copy that very easily and look at the first step in actual copying it, called DNA replication.